Yeah, I want to introduce to you Zerion and what we are doing and what the Dispersome technology is. I guess it's something a bit new compared to what is established. I will talk a little bit about Zerion Pharma, then a bit of a background, where we are coming from, and then I want to talk about what the entire technology is about. So we are a spin-out company from the University of Copenhagen. I have been employed at the University of Copenhagen as an associate professor. Part of the research was then translated into patents, and one of the patents ended up then to be the foundation for the company Zerion Pharma. We are quite young, so we founded the company together with Ole and two other co-founders in 2019. So we are 4.5 years into the journey. And what we are dealing with is typically quite challenging drug molecules that we obtain from the pharmaceutical industry. Our target really is new chemical entities. A lot of money is spent to develop these compounds, but if they don't dissolve, of course, you cannot develop them into drug products. We have developed this platform that we call the Dispersome technology to enhance the solubility of these polysoluble drugs, and in particularly those where other technologies are not working at the moment. And where do we see an application? Of course, naturally, we want to bring more new drugs to the market. That's why we work with a big pharma, but we could also foresee that a reformulation and improving products could be a nice way to apply the technology, and I have also some examples there. Of course, we have protected the technology with a few patent families, so it would also allow the industry to prolong their IP protection and market access. And then overall, if you think about enhancing bioavailability, you could also think about that it would enable you to reduce the amount of API to obtain the same pharmaceutical effect, of course. So very briefly, what is the Dispersome technology? It is when we blend a small molecule drug with a protein and form an amorphous mixture. And that gives you then what we call a dispersome formulation. So we are using proteins that come from the food industry. In particular, we use beta-lactoglobulin, which comes from Arla Foods ingredients. They are one of the biggest dairy companies in Europe. There are several reasons why we work with BLG. One of them is that we have a really good source for getting the BLG in a very high purity and high quality. And the other reason is that we have more or less worked with every protein we could get our hands on, and BLG seemed to have been working best for most of the drugs we have tested. It doesn't exclude other proteins from working in a similar manner, but we have for now at least focused on BLG as the protein of our choice. This is a short snapshot of how it would look in the lab. This is a formulation from one of our partners, where we get a full solution. There's no particles left. And here we have the same amount of drug as a crystalline material, and you can see it's more or less, it is insoluble, and you get a suspension, right? So now I want to briefly go back to the times when we were still in university, so where we coming from, and why we ended up actually working with proteins as a carrier. So as Simon said, I have been working with more or less every technology that can enhance solubility in some way or another. And then when I decided to continue after my PhD into an academic journey, then I was looking into where are the shortcomings of all these technologies, and is there some way to overcome these? So I want to briefly highlight that there are many technologies out there. Of course, these amorphous solid dispersions are more or less skyrocketing in the last few years, and overtaking also the lipid-based formulations, which were maybe a bit more popular. They're still popular, but in maybe 20 years ago. There's also, of course, cyclodextrins, but they're mainly used for injectables, and then, of course, these nanoparticles. Micronizing, you just reduce particle size, and then we go into this DCS class 2A. People often claim you can have a quite high drug loading in the tablet, but of course you also there need some sort of stabilizing excipients. But you're of course certainly limited by the dissolution rate and not gaining from the solubility. Then you have these lipid-based drug delivery systems. We heard a little bit about, or actually quite a lot already during the talk. I don't want to go in any details here, But what we have seen here also is that sometimes you need quite a lot of lipid to actually dissolve the drug in the lipid. We have worked with one of our partners, and their best formulation they could come up screening amorphous solid dispersions, nanoparticles, and lipid-based drug delivery systems was a 1% drug-loaded soft shell capsule. And they were then asking us, do anything that gives you more than 5% in the final dosage form. So it can be also quite challenging, but there are, of course, products. And then what you typically also do are these ternary phase diagrams, because in the end you want to make sure that once your lipid-based system is digested or diluted in the gastric juices, that it doesn't go through several phase changes, which can, of course, also induce crystallization and so forth. And then I have done a lot with amorphous solid dispersions or amorphous drug systems. Also here, there are some challenges with drug loading. This comes all back to this little figure on the right-hand side. It's a phase diagram of drug solubility in the polymer over different temperatures. You can use that also to determine what the solubility of the drug in the polymer is. But as you can see, the solubility of the drug in the polymer is actually quite high when you heat it above the glass transition temperature. But once you cool the system down, you may go into some sort of oversaturation. And what you typically want to do is that you want to stay here on the left side of this red line, because then you have a thermodynamically stable amorphous solid dispersion where you stay below the saturation solubility of the drug in the polymer, even at room temperature. But I think it becomes quite clear that the drug loading is quite low. In our experience, it's usually below 30 weight%, but often we get approached by partners where it's even lower, maybe 10% drug loading. So that's where we, in the academic setting, then looked into what can we do to increase, for example, the drug loading, and how can we potentially also increase solubility. And that's where we tried whatever, so many carriers, everything we got our hands on, and then somehow these proteins seem to work really good. Then we founded the company. Of course, then we scaled up our investigations. We have now worked with more than 50 drugs. Initially, we have worked mainly at very high drug loadings. You can see here, for these 55 drugs we tested, we have quite a high success rate at a drug loading of 50 weight%. We manufacture this via spray drying. We have entered a collaboration with the company Hovione in Portugal, that you may know. It's one of the biggest CDMOs with regards to manufacturing clinical but also commercial batches with spray drying. So you may as well go to a conference and meet Hovione and then also come across the Dispersome technology. That's, of course, through our collaboration with Hovione. And then we have done numerous PK assessments in animals, and we will also see one of them later. So we often get asked, of course, at the moment when we founded the company, we had only little insight into how the technology actually works. We knew it works, but we didn't know precisely how it works. So what we have done is we still have collaborations with both the University of Copenhagen, but also some other universities. This was in collaboration with Uppsala University in Sweden. So we wanted to know what happens, how does it work? Is it similar to how polymeric amorphous solid dispersion works, or is it a different concept behind it? So we did a lot of experimental data. We spray dried several formulations at different drug loading, and then we did molecular dynamic simulations to see where is actually the drug, how is it interacting with the protein. And what you can see here on the left, we prepared different drug loadings here. We put them on storage for 12 months under dry and humid conditions, and you can see at above 50 weight%, the formulations start to crystallize here in the humid environment. If we look at the molecular dynamic simulations, then of course you have your protein structure, and then we add more and more of the drug molecules into the simulations, and we wanted to understand where is actually the drug. So the drug molecules are these blue and red spheres, and you can see that when you start out at low drug loadings, the drug actually wants to be on the surface of the bigger protein molecule. and then it has some sort of molecular interactions. These could be of ionic nature, they could be hydrogen bonding, they could be pipe-high interactions, all sorts of different interactions, but the key message is really that the drug molecule wants to sit on the surface of the protein. So then when you add more and more and more of these drug molecules, of course, eventually you will have filled up all these binding sites on the protein surface, and you see these red spheres appearing, which are drug molecules that have no direct contact with the protein surface. And then, of course, if you continue adding, you get more and more of these red spheres. In the simulations, this seems to be around 35, 50 weight percent, roughly. If you were wondering why these numbers don't add up to 100%, the rest of the molecules in the simulations are water, because typically amorphous systems have at least some water in the formulation. And then, in the experiment, there seems to be a cutoff around 50 weight percent. We have done a few more studies, and now I have made a new figure that maybe summarizes roughly what is happening here. So if you keep your drug loading below what is called the first drug shell, so meaning the drugs that are direct contact with the protein, then you get a stable system that is stable under dry and humid conditions. If you add more, then you can see the proteins also somehow form a matrix where there is some void space in it. You can fill up that void space, and then you still have protein-protein interactions forming a matrix. That goes up to maybe around 80 weight percent. You get still a stable formulation in dry conditions, but you may induce some crystallization when you store it in humid conditions. And what we typically see is if you go above 80 weight percent, then of course the continuous phase becomes the drug, and then you remove these protein-protein interactions, and the system becomes quite unstable. That's why we can actually formulate quite stable formulations also at very high drug loadings. Then we have also recently submitted a paper where we looked a bit more into the mechanism, how the solubility enhancement is actually achieved. Of course, a lot happens when the material is already amorphous. You have a higher solubility than the crystalline drug, but also the protein has a quite high solubility, which, of course, as a carrier will enhance the solubility of the drug if it carries it into the solution. And then you can see here is a model drug which is called furosemide, that the solubility enhancement is somewhat different depending on the pH. Furosemide is a weak acid, meaning that it starts to become ionized at a pH above 4.5 roughly. So the squares are the solubility of the crystalline drug, and the circles are the solubility of the dispersome formulation. And you can see that the solubility enhancement compared to the crystalline form is more pronounced when you have an unionized form, which is also then on the right side related to that the drug actually likes to interact with the protein in solution when it is in the unionized form. There comes also on top of it that once the furosemide becomes ionized at higher pH, it has a negative charge, and the protein BLG has a isoelectric point also around 4 to 5. So also the protein becomes negatively charged. So the interaction between drug and protein in solution, you could say there's probably some repulsion between these two. But you could say the solubility enhancement comes from the high solubility of the protein, the amorphous nature, and the interactions of the drug and the protein, not only in the solid state, but also once it goes into the solution. Here is a comparison to these polymeric amorphous solid dispersion. This was a project that we have done together with a big pharma company. They came to us saying that we have this drug that has really poor solubility. Can you do something? On top of it, they also said it's a high-dose drug. We need something with a high drug loading. You can see we have worked also with the polymeric amorphous solid dispersions with HPMCAS, but also with PVPVA. You can make them amorphous, however, only up to a drug loading of 20 weight percent. Then they start to become physically unstable. We made a formulation here with our BLG at a 50 weight percent drug loading, and not only was it stable, but also had a higher solubility and dissolution rate. Then we have another study that we actually published. It shows more or less the same picture, except that we used also hydroxide L as one of the polymers and also did a nanocrystalline formulation. Not so surprising, the crystalline formulation doesn't get you higher solubility, but the picture is essentially the same. Then another study that we did with the company Bayer. In Germany, they wanted to test the technology and gave us this drug nimodipine that is, of course, one of their in-house drugs. And they said it's quite a challenging drug, even for polymeric amorphous solid dispersions, whether we can do something. And we did a lot, and what we realized is that this is a quite challenging drug, that even at higher drug loadings here, 40%, 25 weight%, there is not so much gain in the solubility of the drug, not from the amorphous solid dispersion with the polymer, but also not from the dispersome. Then we were putting our heads together and say, how is this possible? And then we tried a few other drug loadings, a few other approaches. And you can see suddenly when we go down to 17.5% drug loading, then we get this really high increase in solubility when we do the dispersome formulation. That, of course, we haven't further investigated the precise mechanism, but what I hypothesize is that nimodipine potentially is one of the drug molecules not interacting too well with the protein, or you're filling up the binding sites too quickly. And when you have too much drug in there, we know that also from Lynn Taylor's work in Purdue, then you're basically reducing the dissolution rate because you enrich the drug at the surface of the particles. Yeah, we were quite happy to see this really high dissolution at a lower drug loading. And of course, it would have been nice to follow on with this work into a PK study, how this actually translates in bioavailability, but we haven't done that so far. But what I want to say here is that often it may not be the target to get the highest drug loading, but you also need to compare what you get in dissolution. And then maybe for this particular drug, the most ideal drug loading is below 20 weight%, and it certainly has a higher solubility than what you would obtain for polymeric amorphous solid dispersion. Then this is something we have also worked on quite a lot because we have been facing these challenges that we may not be able to get high drug loading and high solubility, as just shown with the nimodipine example. We have tried a lot to combine the protein with other excipients. And you can see here, we did a combination of a polymer and the drug together. And that gave us a synergistic effect. So it's not an additive effect that you see the dissolution suddenly in between what you would get for the polymeric AST and the protein AST, but they can be synergistic and you get an even higher dissolution and solubility rate. So a lot of the projects we are working on, we are actually not only using the protein, but combine it with other excipients to further enhance the performance of the dispersome formulations. Then we have done a lot of tableting. So what are you doing once you get the spray-dried powder out? I think the process of what is shown here is not so surprising. Spray-dried powder is usually quite fine. So we go through a dry granulation step and then add a few more excipient to get into a tableting stage. I think most importantly here is that we have been showing for some of these examples that we can actually load up to 80% of the dispersome formulation into a tablet and still have quite good disintegration and dissolution behavior. So we are certainly on top of also the tableting work here. Then once you have a tablet, you want to test it in animals. This is data from a dog study on a drug called abiraterone acetate. This is one of the programs where we also want to go ahead with a 505(b)(2) registration process, simply because the technology itself is not yet marketed or even approved by the regulatory authorities. I think we had already some discussion on that yesterday also with a 3D printing approach. So you need to go through some sort of product registration to get the platform approved by the authorities. And this is one of the products we are working on. We made a formulation with high solubility, formulating it as a dispersome formulation. We knew it already has a higher solubility than the marketed product Zytiga. So our target was to reduce the dose to a quarter, which would allow us to go from 4 tablets a day to 1 tablet a day. And when we put a quarter of the dose from the originator into the animals, you can see we got actually a 10 times increase in the bioavailability. So even though we are dosing only a quarter of the dose, the bioavailability in dogs is much higher. Good. And then another project we are working on here is in the cannabinoid field. We've already heard yesterday that one of the marketed formulations is a solution in oil. So cannabinoids are very soluble in oil. But what we were working on, we were interested in, can we transfer a liquid formulation into a tablet formulation that is solid, but still has a high drug loading so that you remain at 1 tablet versus 1 capsule. And you can see here on the left, this is dissolution studies. The crystalline cannabinoid is more or less insoluble. In the formulation with the Dispersome, we get actually 100% release in the red profile here. And then we gave this red formulation into a rat study and compared it to 3 oil-based formulations here, or nano-emulsified, then the marketed oil formulation, and then also one that was diluted because we were pre-suspending, of course, our Dispersome formulation in the rat animal study in a carrier before we were dosing. And we were quite happy to see that our solid formulation gave us a bioequivalent formulation. However, the variation of the PK data was slightly lower. Good. Then I need to talk a little bit about the regulatory status of the BLG, but I don't want to talk too much about it. What I can say is that it is a novel excipient. It is not yet approved as any pharmaceutical excipient yet, but it has GRAS status in the US. It is used, of course, heavily in different types of products. It comes from the whey stream. You may have heard of whey protein isolate, which is consumed for example, by infants in infant nutrition, but also by bodybuilders. There's quite a lot of historic use of milk and milk proteins, and we have already had meetings with the FDA and also with the regulatory authorities and with EMA. They have not shown any concerns for the pilot studies in the clinical trials that we have lined up. So, but we of course also collecting the necessary documentation for filing for this drug master file type 4, but also the excipient information package in the EMA at EMA. Good. So how do we work? So this is a rough overview of our partner structure. We are, of course, having a license to the first patent that we filed together with the University of Copenhagen. We have also a license to another patent family from the company Rousselot, also dealing with protein drug amorphous solid dispersions. Then our supplier of the protein is Arla Food Ingredients in Denmark. We have a strategic partner with Hovione that can take on the GMP manufacturing, the commercial batch manufacturing, but also the commercialization of the technology. And then we have quite some collaborations with the pharmaceutical industry. A few of those are public, like Insud Pharma, Bayer, DSM, and Novartis, and we have also some undisclosed partnerships. To sum up, I think we have in the past 4.5 years shown that the technology is quite diverse and can be applied to many very challenging compounds. Those were the industry has tried everything and it doesn't work, and then they come to us. Of course, we want to bring those drugs closer to the patient. And I can say we have worked with a lot of different compounds, also to higher molecular weights up to 1,000 grams per mole. We want to work with more, even higher, but of course we need access to those type of molecules. We are collecting quality and safety documentation for the excipient registration. We have 2 clinical trials on the way or scheduled for end of this year or early 2024. And then we can also manufacture them up to commercial scale through our partner at Hovione. And of course, I can only say this again, we really have this ambition to bring more new drugs to the market together with our partners. But we can also apply it in a reformulation context where we improve products that maybe are not yet ideal in terms of too much API. Yeah. Thank you. If you have any questions, please let me know.
Thank you very much, Korbinian. Thank you. Yes, questions. Got a few, actually. I'll start, Karu, then I'll work my way this way.
Thank you. Great presentation, and looks like some breakthrough new excipient to improve the bioavailability. So can you just give some color on what the cost of this protein is, or at least some comparison versus the standard polymer excipients that we currently use?
There is no price on the excipient yet, but of course we are aware that we are in competition with other excipients out there, but we have not put a price tag on it yet. But it will be most likely in a similar ballpark. Yeah.
Okay, thank you.
Hi, very interesting talk. This is more out of curiosity and my lesser knowledge on the BLG protein, but in general, proteins tend to denature when they're not, used in optimum conditions. How does this technology work? Whether if you use high temperatures, low pH. I remember you also showed a trace where you looked at the solubility of a drug in different pH environments. And something that I noticed, which again, out of curiosity, I'm asking this question, at lower pH, it seemed there was less solubility, and it increased when it was towards more neutral conditions. Would you say is the low solubility because of denaturation of that protein or otherwise?
I think there was a very specific profile for the model drug that we have been using here. There was an acidic model drug. We have also used some basic model drugs. Then you could say you switch around the solubility profile, because then it turns around and you have high drug protein interactions at a higher pH compared to the low pH. So maybe don't take this particular example too literal here. In the paper that we have submitted, we have actually used 4 different types of drugs. So it is dependent on the drug itself, but also on the BLG, of course. And with regards to the denaturation, it's right. Proteins tend to be quite unstable, let's say, especially in solution when you heat it up, right? But if you have the dry powder, it's actually quite a stable material. We have another publication on the way where we actually look into the structural changes of the protein upon processing. Maybe without saying too much, is the powder, when we redisperse it, it looks all the same as the native protein, right? But there may be some minor structural changes to the tertiary structure upon processing, but these are reversible. Yeah.
Thank you.
Thank you. I'll work my way around to you, Bob.
Hello. So very nice talk. My question is sort of related to that. So you're doing a spray drying from a common solvent in which the protein and the drug is soluble?
That's true. Yeah.
Okay. And so have you then looked at other proteins that might be in the future admissible for parenteral administration, like human albumins or things like that?
Very good question, yeah. We have been thinking about this a lot. We have some data on HSA and BSA from the very early days, so we know it works with these two. But in the end, we have been focusing mainly on the oral market. And then due to our good relations to Arla and the supply chain that we foresee with them, we have put all our focus, for now at least, on the BLG. But the principle itself is you can extend it also to other proteins, and I agree it would be really good to have also one that could be used for parenteral formulations.
Hi, very nice presentation. Just a quick question. Do you anticipate any immune response because the material you're using is more of biological origin?
Well, based on the historic data, there shouldn't be too much of an immune response if you give it orally. but there are, of course, individuals that may have a milk allergy related to the proteins. We have also, of course, looked into the guidelines what to do if you do something like that, but that is typically handled by a labeling on the drug package that it contains milk proteins. Patients with milk allergy should not take this product, right? But there's actually not too many people with milk allergies in the population.
Okay, my question is actually, many of the questions that I wanted to ask were asked by the prior people. But one question, you know, the confirmation of the tertiary structure is very important. Now, if in any case that is, you know, due to denaturation, and after that, do you see any phase separation with the API?
You mean in the solid formulation?
Yeah, you know, for some reason, if pH or moisture, you know, heat, if that changes the conformation, would you see phase separation of the API out of the polymer or out of the protein?
Could be. We haven't looked for that particular scenario yet. But of course, if the changes due to pH or whatever it is, changes the interaction of the drug to the protein in solution, it may induce some sort of precipitation. That could possibly be true, yeah.
Yes.
Very nice talk, thank you.
Yep.
The question related to previous question about this common solvent. Could you give an example of indomethacin or furosemide? What is the solvent composition?
It's a bit of a business secret what we are doing, but I think we have screened, of course, a lot of solvent and solvent combinations that are mutual solvents for both the protein and the drug. And it has been quite challenging in the beginning. I think now we have somewhat figured out how to do it. From all the drugs we have worked with so far, there was only one single case where we couldn't find a common solvent. But it is typically combinations of not only one solvent, but several solvents. Yeah. Yeah. There are also organic solvents that can dissolve proteins. But in some cases, it's possible to make, let's say, an ethanol water mixture. But it is very rare that you find this solvent combination working for the drug. Yeah.
Yeah, my question actually, a good talk actually, I missed, a few slides earlier. Question actually regarding the solubilizers, and when you're doing the spray drying with protein, have you tried some solubilizers with, copovidone, the HPMCAS, and what are the impacts actually on those proteins?
You mean if we make a ternary formulation protein, drug,
exactly.
and polymer?
Yes.
They're usually blending together quite nicely, giving you like some sort of like an homogeneous amorphous phase with a single glass transition temperature. We haven't done any like mechanistic or molecular study into how it looks like in the formulation, but for now I can only say that it is a homogeneous mix, and the combination somehow gives a synergistic effect, but I don't know why. Right? Yeah.
Hi. So the question around, we saw the data that Dispersome has, let's say, the highest release compared to polymeric amorphous solid dispersants. What do you think is the mechanism involved there? Like why you get this boost in the release?
So with the polymeric amorphous solid dispersion, it's of course also with the Dispersome very much drug dependent, right? Ideally, you target some sort of drug loading, and here I need to echo what the work of Lynn Taylor has shown, right? Where you get the congruent release, where the polymer and drug are released at the same rate, because if you have like a too high drug loading, you would release the polymer but not the drug, and then you surface enrich the drug on the particle and get a slower drug release. For the proteins, As we speak, one of our PhD students is doing the analysis whether we also get the congruent release, whether the BLG together with the drug is released at the same rate. I haven't seen the data yet, so I cannot answer it, but I would imagine there is something similar behind it. But in terms of the stabilization in the solid state, it is somewhat different because with the polymeric amorphous solid dispersions, you're looking at miscibility or solubility of the drug in the polymer, whereas with the proteins, we are looking more onto absorption or interactions of the small drug to the big protein surface. Yeah.
Okay. Any last quick questions for Korbinian before we have a break? No? Okay, let's give Korbinian a round of applause. Thank you. Bye. Bye.